Hello, everyone. Welcome to the MSK Science Spotlight series. Thank you all so much for joining us. My name is Omar Abdel Wahab, and I am an associate member in the Human Oncology and Pathogenesis program at MSK within the Department of Medicine. This seminar series features lectures from today's leaders in basic and translational biomedical science. If you have questions throughout the talk, we're going to have a Q&A session towards the end of Charles's talk. And you can email any questions you have throughout the talk to the mskcc.org email address you see at the bottom right-hand slide, or you can tweet your questions to the Twitter handle you see there at MSK Education. So it's my pleasure to introduce our speaker for today, Dr. Charles Sawyers. Charles is an investigator of the Howard Hughes Medical Institute and the Marie Jose and Henry Kravitz Chair in Human Oncology and Pathogenesis. And he's also the chair of the Human Oncology and Pathogenesis program. For many of you, Charles needs no introduction, but suffice to say, he's been a pioneer in the study of signaling pathways that drive the growth of cancer, and his work has directly led to transformative new therapies for patients with chronic myeloid leukemia and prostate cancer, amongst other cancers. Charles's talk today is titled Lineage Plasticity in Cancer Drug Research. Charles, it's my pleasure to introduce you. The floor is yours.
Thank you, Omar, and thanks to all of you for tuning in. Before I get into the meat of the talk, I want to sort of set the stage. Like Stephen Colbert, I'm broadcasting from home today. To prove it to you, I'll show you that my home office is actually the kitchen. You can see the stove over there. You can see the living room over there where we watch our Netflix videos when we have nothing else to do. To get started, I'm going to talk about lineage plasticity, which is a property of normal cells. And I'm going to show you some examples of how cancer cells can subvert these normal developmental programs, these plasticity programs, to not only initiate disease, but also to develop resistance to drugs, in this case, hormone therapy. I'll first show some disclosures. They're listed here. None of these have any relevance to the material I'm going to present. So I want to open with this overview slide, which I'll also close with it at the end. It just sort of frames the talk. And I'm going to just give the talk in 3 sections. I'm going to start with an analysis of the normal mouse prostate gland using single cell analysis, which reveals a lot of complexity in the gland that we didn't previously appreciate. Then I'm going to move to a story about drug resistance, in this case, resistance to hormonal therapy, and how in a subset of patients that can occur through a change in identity, that the luminal epithelial cell undergoes an identity change to escape dependence on the androgen receptor. Now I'll close with some recent work on an oncogenic driver in prostate cancer, FOXA1, and how that also works by initiating a plasticity change, in this case, luminal development. I'm going to acknowledge the people for the first 2 stories right on the first slide. On this first slide, the real hero of the story is Wouter Karthaus, shown here, together with Jimmy Zhao and Samir Zaida from my lab on some of the later work. And we've done this work collaboratively with Donna Pear's group, and specifically Joe Chan, as well as Aviv Regev's group and Matan Hafre. And some of the mouse work I'm going to show you, which is unpublished, is work done collaboratively with David Goodrich and Christine Wadowski at Roswell Park. So I'll begin actually with a patient. So this is a man who was seen at Sloan Kettering a couple of years ago, shared this, the Yu Chen and Anu Gopalan colleagues at MSK shared this with me. So this is a man who underwent bilateral biopsy of the prostate gland for a suspicion of prostate cancer. And what you can see is 2 different histologies. This is a classic adenocarcinoma on the right, and this is read as a small cell histology on the left, which is quite unusual. Nearly all prostate cancers are adenocarcinomas. And what's shown below is the sequencing analysis of both of those independent biopsies. And what they reveal is the presence of some somewhat canonical mutations in primary prostate cancer, such as the Tempers-Herd rearrangement, P10 deletion, but also a mutation in P53. But what I want you to appreciate is the tumor on the left has exactly the same genomic profile, but with the addition of a deletion in the retinoblastoma gene, which, as you will see, sets a genetic context in which this tumor cell can acquire plasticity and undergo a lineage shift to this small cell histology. So with that as a framing slide to sort of put the clinical importance of this problem right up front, I'll go back and review a little bit of prostate cancer biology. So normal prostate is composed of luminal epithelial cells and basal epithelial cells. In a minute, when I get into single cell data, I'll reveal another layer of complexity. But the key point here is that prostate cancers are luminal adenocarcinomas. They have luminal histology when they initially present, and they tend to also retain that luminal histology when they develop what's known as castration-resistant disease, with the exceptions of cases like the one I just showed you. And the types of therapies we use to treat these patients when they do develop metastatic disease are all directed at the androgen receptor. And there's different flavors or generations of therapy that are listed here. And if you think of the sort of life cycle or course of a patient with prostate cancer, the stages of treatment initially are surgery and radiation therapy for localized cancer, and then hormone therapy for metastatic disease, usually starting with primary therapy and then moving on to next generation therapy. Although, as I've indicated in the green arrow, we're rapidly moving these much more potent drugs up front or earlier in the course of treatment. So the first part of the talk is to go into our recent single cell analysis of the normal prostate gland. And this is the work that I mentioned is in collaboration with the Regev lab and the Pear lab. So what this is a TS&E plot, a standard way in which single cell data is represented in two-dimensional space. And the main point I want to make here is that there's lots more cell types than in that cartoon I just showed you. But in particular, there are, if we now shrink that and I drew a little line around the 3 different luminal types of cells, which we have just named luminal 1, 2, and 3. If we then separately run a t-SNE map against just the luminal cells, we see these large luminal population called luminal 1. And these represent, obviously, the dominant population. These have the markers of secretory luminal epithelial cells. And in the human version, these are the classic ones that express prostate-specific antigen. And then I also want to call your attention to these 2 smaller subsets, luminal 2 and luminal 3. Luminal 2 is particularly interesting because it expresses some canonical markers of stem cells from other tissues like SCA1, and also in the prostate, a gene called PSCA, which stands for prostate stem cell antigen. And then luminal 3 actually is one that we never even imagined existed in the prostate. They're defined by a transcription factor known as FOXA1 that was reported by Aviv Regev's lab a couple of years ago in the mouse lung. And it defines a cell called an ionocyte, which is involved in ion transport and is important in the phenotype of cystic fibrosis in the lung. We really don't know yet what it's doing in the prostate. This slide shows that those findings from single-cell RNA-seq can be reproduced through in situ analysis using immunofluorescence of antibodies to the different markers, and they define anatomically the position in the prostate gland of the L1 and L2 components. And the main point I want to make here, I'll draw your attention to this portion on the bottom right, in which you see red, which is marking luminal 2 cells, suddenly flipping to a greenish color, which is luminal 1 cells. And that represents a fairly abrupt transition as you move from the proximal part of the duct to the distal part of the duct, suggesting that there is probably some role for the surrounding microenvironment or supporting cells in specifying the luminal 1 versus luminal 2 fate. What I'll now tell you about is further extension of the single cell analysis to ask what happens to these different populations when we put the mouse through a hormonal manipulation known as castration and then regeneration. And this is a procedure that was developed decades ago to make the point that in the normal mouse, the prostate gland completely involutes upon castration, but then completely redevelops into a normal prostate gland within 28 days after supplying androgen back. And you can see that dramatic change in histology down here. You see the lumen with all the secretions in the normal gland. You see all of that collapse during castration at 4 weeks. And over here, you see all of it coming back 28 days later. What I want to call your attention to on the top is the pink staining, which is staining for Ki67, a proliferation marker. What I hope you can appreciate is that there are proliferating cells in the intact prostate, but it's rare, just a couple of percent. Whereas after just 3 days of adding androgen back, essentially every cell in the epithelium is now pink, meaning there's a massive, brisk proliferation that then dies off as the gland regenerates. So in order to understand this in more molecular detail, we did single-cell RNA-seq profiling of all these different time points shown on the arrows at the top. And there's a lot of data, which I'll summarize quickly in what's called as a fate graph. And these are a little complicated, and I don't have time to go through all of it. But what you see is sort of a wheel of data in which each color represents a time point on the left. And the main point I want to make from the time points is we start over here, the intact mouse, and during the phases of castration back through regeneration, the transcriptomes of the luminal cells assume their original position, but they undergo quite dramatic changes during the cycle. On the right, in the middle section, is dividing that same plot and scoring it based on whether those cells represent luminal 1, luminal 2, or luminal 3 signatures, cells based on the signatures from the intact gland. The first point I want you to see is the green cells, luminal 3, don't change. Although they are androgen regulated and they have androgen regulated gene expression, it's not the most robust part of their transcriptome, so they don't shift in position dramatically. The L1 cells shift the most dramatically, but L2 cells do as well. And what you can also see is that in this portion over here, the transcriptomes of the L1 and L2 cells start to overlap. In other words, L1 cells become L2-like as they undergo the castration response. On the bottom is now another way of annotating the proliferation response, in this case by measuring message level for Ki67 or for various cell cycle markers. And again, what I showed you before is happening here, and that is within 2 days of regeneration, 68% of the L1 cells are back in cycle, and 45% of the L2 cells are back in cycle. So this is a remarkable recruitment of a lot of cells all at once into cycle, and it raises some questions about a concept that's been out there for decades, and that is that there should probably be a stem cell in the prostate that is responsible for regeneration. But this result would suggest that, in fact, it seems that most of the persisting cells, if not all, are responsible for regeneration. This is further Ki67 data showing, which is a bit too faint, I think, for you to see, but again, the brisk Ki67 positivity within 2 days of regeneration. So this slide now moves to a functional test of regenerative capacity. And we can do this in 2 ways. One is by organoid culture, where we can seed single cells into certain conditions in matrigel, serum-free, that allow them to grow as organoids. And you can see pictures of these organoids down here on the bottom. And the first point I want to make is that you can measure the efficiency, the percent of seeded cells that form an organoid that looks like one of the ones below in this bar plot. And about 4% of luminal 1 cells will form an organoid, but about 8 to 10% of luminal 2 cells will form an organoid from an intact mouse. And this fits with the notion that the luminal 2 cells, which if you remember, were the rare subpopulation in the intact gland and express SCA1 and other stem cell markers, they naturally have more regenerative capacity. But what surprised us is that when you look at the red bars, this is the same experiment, but now isolating the luminal 1 or luminal 2 cells from the castrate mouse. And you can see there's almost more than doubling of the capacity of a luminal 1 cell to form an organoid. Luminal 2 cells also change, but not significantly. And over here on the right is looking after regeneration has started at day 2, when all those cells are proliferating, and now the scale on the y-axis is different. You can see that luminal 1 cells now, 1 out of 5, or over 20%, are forming organoids. So that's a remarkable, at least 4 to 5-fold increase compared to baseline over here. So there's something about the castration process that's resetting a self-renewal program in these cells. So the second way we can look at this is through lineage tracing. And we took advantage of a mouse that others have made called the confetti mouse, in which 4 different fluorescent markers can be activated when you turn on an inducible Cre allele. And Wouter cartooned this out here with a little sense of humor on the 2 possible models. So the first model is that there's a certain number of cells persisting post-castration, and we can mark them all with this color. And if every single one is contributing, well, that's sort of the communist model in which all the cells contribute to the regenerated gland, versus a stem cell model in which a smaller subset of cells would contribute to the gland. So what's the answer? Well, it's shown here, and it's the communist model. So let me walk you through this. So here's the way the experiment is set up. We use a luminal specific inducible Cre marker, which we've crossed into this confetti locus. We then castrate a mouse, wait till the gland is fully regressed. Then we add tamoxifen to mark the residual cells. And you can see over here that we successfully marked cells. You can see lots of little colored dots. Then we add testosterone back to regenerate the gland and harvest the gland now 4 weeks later when it's fully regenerated. And you can see here lots of small dots of colors, which when counted more carefully and looked at in higher power, each represents on average somewhere between 2 to 4 cells per clone, reflective of 2 or so doublings over that period. And our conclusion is that there's an orchestra of regeneration that contributes to the regenerated gland. So with that as the backdrop, I want to now turn to cancer, which is sort of the second vignette of the talk. And to set this up, I want to give you a little view of the genomic landscape of prostate cancer in 3 different stages. The first is local regional disease, which is primary cancer. And in the middle is metastatic disease that has not been subjected to hormone therapy. And the third column on the right is castration-resistant metastatic disease, the lethal phase of prostate cancer. And what I've done intentionally is drawn a red box around the frequency of alterations in 2 genes, P53 and RB1, which I mentioned in that opening patient presentation. And I'll call your attention to that because these 2 genes have the frequency of mutations in these 2 genes is tremendously enriched as you go from primary disease to late-stage metastatic disease. I will point out that those of you who are carefully looking might notice that there's 27% frequency of P53 in this local regional plot. This actually represents a high-risk, locally advanced disease group that was part of this initial MSK sequencing cohort. Larger studies of primary disease would put the P53 mutation rate closer to 8 to 10%. But in any case, the point is there's tremendous enrichment. So a couple of years ago, Ping Mu in my lab, a former postdoctoral fellow who now has his own group at UT Southwestern, did this experiment. He took that human data and asked the question, if we delete or impair the function of either RB or P53 alone or in combination in a human prostate cancer model in which both genes are intact, and the one we used is listed here called LNCAP-AR, as well as another one called 22PC, both of which are wild type for those two genes and which are display different degrees of sensitivity to androgen therapy. What he shows here is that when you delete either one or the other by itself, you don't really impact the sensitivity of the cells to the antiandrogen enzalutamide. But when you impair both genes at the same time, you develop resistance. And probably the simplest way to point that out is this pink line here, in which you see tumors growing through the treatment of enzalutamide that have knockdown of both RB and P53. And if you want to get into the finer details, of course, this was published several years ago. Now, a really critical point about this, let me just go back about this observation, is that when Ping looked at the activation state of the androgen receptor, meaning expression of genes like prostate-specific antigen and others, he found that they were still suppressed, despite the fact that the tumors were growing. And that was a bit of a head scratcher for us, because every other time we'd seen resistance to enzalutamide, we'd seen restoration of androgen receptor signaling. And in general, that's what you see in patients when they initially relapse on hormone therapy. So in order to sort of get to the bottom of that mystery, Ping looked at the expression of various lineage markers in these cells. And what you can see here is that the expression of certain luminal markers like cytokeratin 8 and the androgen receptor go down within, in this case, 48 hours of inducing the hairpins that lead to the dual knockdown of RB and P53. And that's further illustrated by RT-PCR analysis of these various lineage marker genes shown along the bottom: basal, neuroendocrine, and luminal. The take home here is that the luminal gene expression tends to go down and basal gene expression tends to go up. Again, across the population within a very short time span, just 2 days after inducing the knockdown of these two tumor suppressors. And finally, he was able to show that this also correlated with resistance to enzalutamide, as shown over here. And what's in the paper, and I won't take the time to show you, is he could actually wash out the doxycycline, watch the genes go back to their baseline state, and see that enzalutamide sensitivity was restored. So unlike the usual form of acquired resistance that at least my lab has been used to working on, in which mutations in a target gene or in a related pathway lead to a fixed state of resistance, in this case, it's reversible. So what's the mechanism behind this? Well, in this particular set of models, we showed that SOX2, the master regulator, reprogramming factor, one of the key 4 Yamanaka factors for reprogramming of fibroblasts to embryonic stem cells, SOX2 is critical for this process. What's shown in the top is the induction of SOX2 upon double knockdown in this model. And what's shown in the green is that if you impair SOX2 knockdown, you see that the induction of basal genes is now blocked. And finally, over here on the top right, you see the induction of drug resistance, which I showed you earlier upon knockdown of RB and P53, is blocked when you also knock down SOX2. So that says that SOX2 is necessary for this lineage plastic state. What about when we look in vivo? Again, it's necessary, as seen by this green sort of triple knockdown experiment. But importantly, the purple, which you can see sort of along the bottom, in which we asked a different question. What if we leave RB and P53 in their wild type state? Instead of knocking them down to get SOX2 up, what if we just overexpress SOX2? Is that sufficient to cause castration-resistant growth? And the answer is no, it's not. So the puzzle is more complicated. So a couple of years ago, when we published that paper, we sort of painted the picture this way in this cartoon. If you're a wild-type tumor for both of these tumor suppressors, you've acquired a fixed commitment to the luminal lineage. And if you're hit with antiandrogen therapy, which targets luminal cells because androgen receptor is a survival factor for those cells, you can't escape unless you somehow bypass the blockade by restoring the pathway. But if you've deleted these two tumor suppressors, you've now, by activating SOX2 and other genes, you've acquired another option, which is to change your identity. In this case, escape by no longer requiring the androgen receptor because you're no longer luminal. So I now want to turn to some unpublished data that we've generated recently in a collaboration with David Goodrich and his lab at Roswell Park. And at the same time that we published our paper, David's group published this paper as a companion paper. In fact, both of our groups collaborated on both papers, in which he reported the phenotype in genetically engineered mice of knocking out either RB and P10 by itself, DKO, or RB, P10, and P53, TKO. And you can read the paper for the details, but the main point I want to make is that these mice naturally progress from a luminal adenocarcinoma to a neuroendocrine carcinoma that synaptophyes and positive within about 14 weeks or so. So having gained this experience with single-cell RNA sequencing of the normal prostate, we now wanted to ask, can we track disease progression in this GEM model of prostate cancer, which evolves to neuroendocrine disease? So this analysis was done by Samir Zaidi and Joe Chan in the Paralab with help from Jimmy Zhao, and the mice provided by David and Christine. And this is a small, it's not an enormous number of mice, but there's a clear set of messages that's emerging. On the top, you can see the plots of the different cell populations colored by genetic background. So the green is wild type. The first point I want to make is that there's a cloud of gray over here. And if you look at that cloud of gray, it appears in the triple knockout, and those cells are the neuroendocrine cells as shown here. The other thing you can see, and this color bar is marking expression of GFP, which is activated upon Cre deletion. And you can see this large population of GFP positive cells, some of which are from normal, but a lot of which are not. Their transcriptomes are dramatically changed. And this is an expansion of the luminal population. And then finally, there's a mesenchymal population that is marked with the transgene, which we believe, but haven't yet been able to prove until we do more lineage tracing types experiments, is likely an EMT transition. We can also take the signatures that I mentioned earlier, the RNA signatures of the luminal 1, luminal 2, and luminal 3 cells, and ask in this mouse model, are the GFP positive sort of tumor cells, do they resemble one or other of these different luminal subtypes? And the answer is, they look a lot like luminal 2 cells, which suggests that perhaps luminal 2 cells, those SCA1 positive cells, might be the cell of origin for at least this genetic combination of events. And finally, you can look at these in what's known as a trajectory analysis, and you can see here that the synaptophysin positive cells may be emerging in this sort of trajectory from a luminal population. Again, proof requires lineage tracing, and we're working on those kinds of experiments now. So one way we can get at that without having to make another mouse model is to go back to organoid culture. And this is the work, again, of Wilder Carthouse. And I want to show you how this works by showing you this movie. So this is a wild, in this case, to sort of set the stage for the way the movie rolls out, we've got two alleles, red and green. And when Cre is introduced, the red is deleted and the green appears. So let me activate. The one thing I forgot to check on my Zoom practice was whether the movie made it over. Sorry, false advertising. You'll have to contact me later for the movie. Here I can show it in cartoon form. Here's what the luminal epithelium, a normal organoid looks like. It forms this nice lumen and a rim of basal and luminal cells cartooned out here on the bottom. These organoids were derived from a floxed RBP53 mouse, which did not have a Cre allele in the mouse. We add the Cre exogenously after we establish the organoid growing ex vivo. And then we can see the cells, the lumen fills up with cells by about 4 weeks. And at later time points, we start to see cells migrating outside from the initial ball of cells. And when we do bulk RNA-seq, we see the expression of SOX2, like in the earlier model from the human system, as well as classic EMT regulators like ZEB1 and snail, and then neural genes such as SLT2, SLT3. But these cells do not go all the way to the neuroendocrine phenotype in this organoid culture, like I showed you in the GEM model. However, if we put them back into the mouse orthotopically, which we do here, so we take a hormonally intact male and through a surgical procedure inject cells into the different lobes of the mouse prostate gland. The injected cells are marked by GFP, and so you can see them now integrating, interspersing into the normal gland. You can see that they still express the androgen receptor. We let these develop for about 6 weeks and then castrate the mice, and then wait 2 weeks later. You see a very dramatic phenotype, and that is much more impressive sort of invasion of the green cells into the gland. You also see for the first time, which we didn't see in organoid culture, complete loss of androgen receptor expression, and now it's a bit faint, but the acquisition of synaptophysin positivity, just like in the genetically engineered mouse model. So of course, the beauty of this system is we can control this in a time frame and sort of pick our time points. So together with Joe Manusetti and Donna Pear, Wouter has done this single cell RNA-seq time course. And what I'm showing you in this map with these colors are just the time points. So as you can see over here on the right in blue, that's the normal organoid. And I'll tell you that there's 3 main populations, 2 luminal and 1 basal. I should just clarify, compared to the normal prostate gland, when we grow cells in organoid culture, we're only growing epithelial cells. So we lose all the mesenchymal and immune cells, which is an advantage for some reasons and a disadvantage for others that we can talk about. But the main point here is that after deletion of RBP53, just 2 weeks later, before we see that histologic phenotype of the lumen filling up with cells, we see dramatic changes in gene expression, which progress further over time. So what are those changes in gene expression? So Joe has done a series of exhaustive analyses for various gene sets. And again, at the top of the list are interferon-related pathways and the JAK-STAT signaling pathway. Something I'd love to talk about in the Q&A is why are these pathways being activated? Because there's no cytokines that I'm aware of that we're adding to these cells. And we've talked about this extensively in our group. The cells themselves do not acquire the production of cytokines. So there's some tantalizing thoughts here. One is activation of the cGAS-STING pathway, which could happen as a consequence of the aneuploidy that develops in these tumors very quickly upon deletion of RBNP53. Another way to look at this is at the gene by gene level. And these maps are oriented as the normal cells down here, and then the time course goes up sort of on a diagonal to the upper left. And you can see that genes like STAT1 and these 2 interferon response genes, ISG15 and IFITM3, just go from 0 to very high expression over this time course. So to summarize this second vignette, or really both of them, I've shown you that normal prostate luminal cells have complexity that we didn't appreciate. We've identified at least 3 subtypes. But really interestingly, when you castrate the mouse, the ones that survive acquire this new state of self-renewal and regenerative capacity. And we can look at that by organoid regeneration as well as the in vivo lineage tracing that I showed you. And then the last parts are how that program of luminal differentiation can be subverted by tumor cells in the context of deletion of RB and P53 and evidence as well that PTEN in various combinations with either RB or P53 or all three can all do that. But it appears that you need at least two of the three in order to get that phenotype, but the rules are still being deciphered. We can model this transition in the GEM model, as I showed you from the Goodrich lab, as well as an organoid culture. And then again, this striking finding, I didn't show it in the mouse, but in both models, of these pathways being activated, which we'd very much like to understand and hopefully exploit. So in the last, say, 12 to 15 minutes, I want to end with the third vignette. And now I'm shifting to prostate cancer initiation. And here's another oncoprint map of prostate cancer from the Cancer Genome Atlas. And I've drawn the red box this time around FOXA1. So FOXA1, I think, as many of you know, is famous as a developmental transcription factor important in specification of endoderm. And it's also famous as a pioneer transcription factor that is involved in opening chromatin because it has this property of being able to bind histones directly in addition to its DNA binding motif. Another point here is that this subgroup is distinct from the quite well-known ETS fusion prostate cancers defined primarily by the ERG translocation, which represent over 50% of cancers. So based on this, as well as a lot of other data, there's pretty much universal agreement that these are initiating events and define separate subtypes of prostate cancer. So one of the, and this story is the story primarily led by Liz Adams-Wisher, who's recently left the lab and is now working at Constellation Pharmaceuticals in Boston, and has been picked up by Abbas Nazir, a new graduate student in my lab. And we've done this work in collaboration with Christina Leslie at MSK and her graduate students. And this work was published about 6 or 8 months ago. And so I'll move somewhat quickly. But this map, lollipop plot of the mutations in FOXA1 in prostate cancer sort of presents you with a couple of interesting questions. One is, there are mutations throughout the gene, which in usual circles would mean that you would worry about loss of function or think of that as the primary consequence of mutation. But there is definitely a hotspot, which you can see here, which maps to this region known as Wing2. There's some details down at the bottom about the stage of disease in which these mutations are present. And the take home from that is the Wing2 mutations are primarily in early stage disease, whereas this one rare mutant, R219, is slightly enriched in late stage disease. And I'll come back to that. We can also, through taking advantage of the structure of another FOXA family member that was solved several years ago, map these mutations and also plot them in this way. And here's the take home, that FOXA1 alterations collectively in prostate cancer represent 12% roughly. That includes mutation as well as focal amplification. But amongst the mutations, over half are those Wing2 hotspots. Another quarter or so are truncations. And then the 219 one is much more rare. In terms of where they localize, this blue region here is Wing2, which you can see inserts itself into the minor groove of DNA, as opposed to helix 3, where the 219 mutation resides, which is the main DNA binding part of the protein. The 219 inserts itself into the major groove, and 219 forms a direct DNA contact site. So with this as a background, Liz took advantage of the organoid system to now build a set of inducible cell lines in which she could inducibly express either wild type or various mutants of FOXA1 and score their phenotypes. And I'll show you the first part of that here. So first, here's a normal appearing organoid. One of the first experiments was to delete FOXA1 by CRISPR. And what you can see is that you don't get lumens appearing anymore. the architecture of the gland is mostly retained, and they grow more poorly. And this is entirely consistent with what has been shown years ago by Bob Matusik and others, in which you selectively delete FOXA1 in the prostate gland. Basal cells accumulate and you don't get luminal cells. So now if you overexpress FOXA1 in organoid culture in standard conditions, you see that they grow faster, and that's in red. But under the normal conditions, they grow pretty fast as well. In order to tease this difference apart, we removed one of the key growth factors in organoid culture, and that's EGF. And you can see that now you get EGF-independent growth with overexpression of FOXA1. Using that as a sort of benchmark in red, Liz then scored this large panel of mutants, which were picked based on their relative frequencies and positions in FOXA1 in the human cancer data set. And you can see, with no exceptions, all of them grow faster. So that's quite interesting and really strongly supports the notion that these are gain-of-function oncogenic mutations. There's a lot of busyness on this slide, and I'll refer you to the paper for details. But what's important to point out here is the histology of these organoids is impacted by the level of FOXA1. What I hope you can appreciate is bigger lumens. It's really dramatic here and here. There's more cells, and they retain, for the most part, their normal architecture. But the one exception is this 219, which is the helix 3 DNA binding contact site. And that's, in this case, you get a phenotype that looks almost like FOXA1 deletion, except that they grow faster. And the polarity of these organoids is altered. The normal basal luminal structure does not hold up. So I told you that FOXA1 is famous as a pioneer factor. So one of the key readouts that we wanted to look at was to measure open chromatin as a consequence of turning on either wild type or mutant FOXA1. And this is the work we did in collaboration with Christina's group. And what I'm showing you first is just plotting the number of peaks that are either open or closed compared to control. And the scale here is up to 4,000 peaks. And what I want you to see is that within 24 hours, you can see upwards of 1,500 new peaks with this one and over 3,000 with this one. And if you go out to 5 days, and now we've added a couple of other controls of FOXA1 deletion, you can see over 4,000 new peaks with this Wing2 mutant and over 6,000 with this Helix 3 mutant. Of course, the real question is, what is the pattern of these peaks, and are there any clusters? And the answer is yes. So in this clustering analysis, Christina's team generated, we can make the following conclusions. So what's shown along the top is the genetic context in which the organoid is grown, either with an empty vector, overexpression of wild type, Wing2, Helix 3, and then the last three, this is another control. It's essentially empty vector, but this time with a non-targeting guide. And these are two different guides targeting FOXA1. So here's what I want you to take away first, that by just looking at these last three lanes, you can see that cluster 1 drops out when you delete FOXA1. So we would call these sort of native mouse prostate FOXA1 peaks. And you can see that those are retained, obviously, when you overexpress the different alleles. Over here, you see cluster 0. These are new peaks that appear upon overexpression of wild type or Wing2 mutant. And Wing2 is much more potent at generating, these are larger peaks. I don't have time to show you, but the motif analysis here shows classic FOXA1 binding sites, similar to those here. The real outlier is cluster 3, which is uniquely present in the DNA binding mutant, the Helix 3 mutant, which, if you remember, I mentioned in the clinical context is enriched in late-stage disease. And these peaks do not have the canonical FOXA1 motif, they have a sort of an aberrant motif, which wild-type FOXA1 can bind to some degree, but this mutant acquires a higher affinity for binding to. So we call these neomorphic peaks. To conclude then, Wing2 mutations are the most common FOXA1 mutations, and they appear to be, really hyperactivated versions of wild-type FOXA1, and they drive proliferation and differentiation of luminal cells. The Helix 3 mutants, much more rare, enriched in later stage disease, and I didn't show you, but transcriptionally don't drive an luminal program. I showed you that histologically. It looks more like EMT. And then finally, we think these biologies are driven by their properties in opening or closing different regions of chromatin, and understanding the downstream genes that are activated as a consequence of that is one of the next key questions that we're interested in looking at. So I'll wrap up now by going back to the slide that I started with. I've told you 3 stories of lineage plasticity. In the normal context, I showed you how castration can change the self-renewal properties of a luminal cell that in an intact mouse is largely just committed to secretory function, the L1 cell. In the second story, I've shown you that in the context of RB and P53 loss, as well as P10 loss, luminal cells, and I drew this arrow intentionally to be a bit darker, appear to be the cell that can give rise to this transdifferentiation phenotype. That's based on single cell computational predictions, and we'd love to get that evidence through lineage tracing. Whether a basal transition is possible, I don't know. But finally, in the FOXA1 story, which I should also say, there's a very similar story from Yu Chen's group with ERG. Both have the ability, when expressed in organoids as well as in mice, to drive cells to a more luminal differentiation program and luminal proliferation. So with that, I'll stop and open it for questions. So Omar, welcome back.
Yeah, thank you. Thanks, Charles. That was great. So the questions are open. We've gotten a bunch. People can email them in or tweet them in. We have a little under 15 minutes for questions. Charles, I'm looking at some of the questions. I'm going to ask you a few. First is this observation about this luminal to neuroendocrine transition, with the P53 RB loss. Do you have any ideas if this is happening in other neuroendocrine cancers, like, for example, within the lung or the GI, where a similar sort of observation might be happening, where the luminal cells are giving rise to neuroendocrine type of cancer as well?
Well, in the lung, there is a remarkably parallel story. I don't know that there's a luminal cell in the lung, but it was first actually described by Jeff Engelman and colleagues several years ago clinically. They noticed that patients with EGFR mutant lung cancer who received an EGFR inhibitor, most of the time they relapse with the classic gatekeeper T790M mutation, which I guess osimertinib is now taken care of, but they can relapse with small cell cancer. And initially, it was confusing because the thinking was maybe they got a second cancer, but they actually have the EGFR mutation present. So Charlie Rudin, our colleague at MSK, has reported recently that there are patients with EGFR mutant lung cancer that also have RB and P53 mutation. And those patients have a much higher probability of undergoing this shift. So yes, it is definitely true in lung cancer. I don't know of specific examples in other cancers, other neuroendocrine cancers. But I think the theme of plasticity as a way of escaping targeted therapy, there's sort of footprints of it all over the place. But whether RB and P53 are the key drivers all the time, I don't know.
Got it. Yeah, that's great. Yeah, so several questions related to that with the, and actually something you wanted to talk about further as well, this upregulation of, JAK-STAT signaling and interferon signaling that you're seeing with the RBP53 loss. Some people are wondering, is that immune related? do you have any sense of new therapeutic targets potentially based on those gene signatures that you're seeing? Any thoughts there?
Well, the answer is we're very interested in all those questions, and I don't know the answer yet. But what I think is, I sort of made the point, but I want to really underscore it. We see this in the organoid. We don't just see this in the gem model, where there's all kinds of cell types and inflammation and cytokines and all kinds of reasons that you might see something like this, either as an important part of the biology or as just, inflammation. But we see it in the organoid culture, which only has epithelial cells, right, no serum, matrigel, and just a small cocktail of growth factors. So it's a head scratcher, I think. As you know, our colleague Sam Bakum, made this important observation about cytoplasmic DNA leaking from the nucleus in cancer cells due to chromosomal instability. And these cells have chromosomal instability, but it's been a double of to try to really prove that that's what's the explanation. I'm tending to doubt, based on the amount of elbow grease we've thrown at it, that is not necessarily the explanation. I've seen some data in other contexts where reactivation of endogenous retroviruses can activate this pathway. So we're sort of barking up that tree as well. But I mean, if someone wants to contact me offline and has a better idea, I'd love to hear from them.
Yeah, in fact, someone brought that up in the questions. They were asking, apparently there's some literature about P53 being upstream of some repetitive elements and non-coding RNAs, and if that's involved, I might send you that question. It might be helpful for you, actually.
I'm familiar with some of that literature as I've sort of snooped around trying to find an explanation. Another context in which this is really important is, which you probably know about from your work in leukemia, is when you give DNA demethylating agents to the DMT inhibitors, you can see activation of a lot of these endogenous human retroviruses, and you get an interferon response, and that may actually be part of the reason those drugs work. Through a through a paracrine sort of immune component. So of course, I mean, if this is really what's going on, we're still getting the disease in the gem model, so it's not able to. So the immune system isn't coming to the rescue, but maybe we just need to figure out a way to help it.
Got it. No, that's great. Okay, switching gears to the first part of your talk about this 3 types of luminal cells that you're seeing. I think you alluded that maybe that luminal 2, at least in the P53RB model, appears to potentially be like the cell type of origin for the cancer. Was that right? And do you have any sense of what these 3 cell types are doing functionally?
Yeah. Well, so that illusion was basically saying that a signature of L2 derived from the normal prostate, when applied, when applying a signature for L1, L2, or L3, they look more like L2. So it's a correlation. We'd have to lineage trace, what's very cool is we have the ability now, L1 and L2 can be separately isolated because they have different cell surface profiles.
Oh, that's nice. Yeah.
So we can ask, what happens if you, put that combination in L1 versus an L2? I'll just say not all gem models are L2. Jimmy Zhao, who was on the slide, he's got a set of data with a MYC transgenic model. And in that case, it looks more like L1. Those look more luminal. So, and I guess the last part of that question was, what are the functions of these normally? So L1 cells look everything like the secretory cell that makes all the prostatic secretions. So the most sort of luminal facing cell, what I would think of as the most terminally differentiated, in sort of normal, the way at least I used to think about differentiation. The L2 cells, they look like cells that are responsible for regeneration, maybe for the normal sort of turnover, which is very low in cells. But I also tried to make that point in the immunohistochemistry or immunofluorescence, that they have anatomic locations in the gland that are probably important in terms of how they branch. Sort of reminds me of branching in mammary gland and lactation and so forth. The L3, we had no idea there would ever be such a cell. We'd never even heard of it. It looks just like this ionocyte, these group has described. It turns out it's expressed more in epididymis, whatever that means. It's involved in proton transport. It's a bit of a lift to figure it out, but the mice there are mice that have been made in which you can delete the transcription factor, the master regulator transcription factor, that was made for other purposes involving kidney. So we could ask.
That's interesting. Yeah, I noticed in that bar plot you showed, sorting the individual cells and looking at the ability to form organoids. You showed, I think, L1 and L2, but I didn't see L3 on there. I don't know if you've tried to see if they'll form organoids as well, or.
The reason you didn't see it is because we can't isolate them yet.
Okay.
There's no magic hook with existing antibodies. So the trick there, someone's made this a GFP mouse with that transcription factor, FOXI1. So Jax has it. If we can get that mouse, which you can't get nowadays, we could sort the green cells from the prostate and figure it out.
Okay, last few minutes, we'll ask some questions about the third part of the talk, the FOXA1 portion. So you talked about these differences between the Wing2 and the Helix 3 mutants. Any idea, based on where those mutations are located, how they might be causing these differences in the chromatin peaks you're seeing and functionally differences between them?
Well, the Helix 3 mutant, this 219 mutant, that's conserved throughout evolution in FOXA1, and we haven't directly shown that it mediates DNA binding in our context, but it's almost impossible that it doesn't. So one of the sort of first puzzles when we saw those peaks opening up is, how is that even possible? It can't bind DNA, but it must be binding DNA, and we showed that it does by ChIP-seq. So it's possible, and this is something that Abbas is supposed to be working on, or will when he gets back to work, is that the Wing2 domain, it also has these DNA contact sites. So maybe that's sort of hanging on to DNA through a different way. And as I didn't show you the data, but it's in the paper, the sequence motif in that cluster 3 is a degenerate motif. It's not called by the usual Homer algorithms. They completely missed it. Christina's student actually stared at the motif and goes, wait a minute, that's just one nucleotide off from a FOXA1 motif. And that's what led us to then test that. Why do the Wing2 mutants lead to sort of super function? I don't know. There's one result, and others. There's some other papers that have come out since ours that show that those mutants can bind to the androgen receptor with much tighter affinity. So it's possible they might bootstrap their way to AR binding sites and then open chromatin through that mechanism to get to new sites. But having said that, the FOXA1 motif is there at those Wing2 mutant sites.
Got it. Okay. We have a couple minutes left. I'm going to ask you maybe just some really things actually I was curious about as well. So the 3 types of luminal cells, is that entirely mouse data? Do we have evidence yet in human prostate that similar types of heterogeneity exists?
Yeah, great question. I think sounds like you reviewed our paper.
No, that certainly didn't.
No, we then yes, the 3 do exist in human, but it's a little different. So first of all, by single cell, this was one of the great things about being at Sloan Kettering, because we were able to partner with Brett Carver and others in GU oncology, GU surgery to get primary radical prostatectomy samples and look at cancer as well as normal, including after the men had been treated with androgen deprivation therapy. So the take-home messages from the human are, first of all, there are L1 and L2 and L3 cells. The L3 cells we didn't see by single cell, but we did see them by immunohistochemistry. So they're rare. But within the L1, there's kind of like a sort of intermediate population as well, that maybe it's a bipotential L1, L2. So that's the answer to your question. But the other part, the reason we looked at the patients who had received androgen deprivation therapy, is this this plasticity of a luminal cell and acquiring a greater self-renewal potential after castration, that rings true in human as well. And so, I don't know exactly what that means for the clinical use of androgen deprivation therapy, but it makes you scratch your head a little bit and wondering, we're kind of turning on a stem cell program when we do this. And is that really the best idea?
Excellent. Well, that's very interesting and thought-provoking. Charles, we're right at 5:30. There's a lot more questions, but I think, they want us to finish by 5:30. So I think we can just go ahead, everyone, and virtually thank Charles for an amazing talk. And in addition, I wanted to just make sure everyone's aware that the next seminar in this spotlight is going to be on Monday, April 13th, and Dr. Scott Lowe is going to present. It's going to be at the same time, 4:30 p.m. Eastern time. So thanks everyone again for joining, and thanks to Charles again.
And also, everyone, I'd like to wish everyone a happy Passover. We're going to have our little virtual family Seder in about 2 hours. So
excellent.
Have a nice evening.
All right. Good night. Bye, everyone.